For those of you who don't know me, my name is Dr. Madeline Diaz, and I'm a postdoc here in the MEMI Project to Cure Paralysis. I have the honor of introducing today's seminar speaker. He is a tenured associate professor in the University of Arizona, where he has successfully risen the ranks in the last 10 years. Since his graduate training, he has dedicated nearly his entire research career to the field of stroke, consistently with a strong focus in investigating novel therapeutic targets and preclinical models, using mainly rodents, but also rhesus monkeys. As a postdoc, he developed a novel post-stroke dementia mouse model, and is currently investigating how chronic inflammatory responses in the stroke lesion can lead to cognitive decline. It is evident that he has already made significant contributions to the field, with at least 35 peer-reviewed articles and active NIH funding since the start of his faculty position. So I've actually had the pleasure of meeting him over a year ago after a mutual collaborator introduced us. So despite our diverse research interests with traumatic brain injury, Niemann-Pick's disease, and stroke, we found several overlapping themes. So since then, we have created an active collaborative group where he is a key member among several national and international scientists. And in every meeting, I have found him to be has always shared incredibly thoughtful questions, has engaged in meaningful discussions, and has been transparent about ongoing projects, the good and the bad. So, and we've been there to collaborate to celebrate the small victories and help troubleshoot when needed. So because of this, I'm grateful he has helped our own research make significant progress in such a short amount of time. So in summary, he has grown to be a fantastic, an insightful and generous colleague. So without further ado, please join me in welcoming Dr. Kristian Doyle.
Well, great. Thank you. I think that's the nicest introduction I've ever had. So I appreciate that. Okay. So as you can see, today I'm going to be talking about foam cells and us using them as a target to improve stroke recovery. And I'm going to begin by giving an introduction to stroke physiology, because I was talking to Dan earlier, and I'm not sure that many of the people here are engaged in stroke research. So with that background in terms of stroke, I study ischemic strokes. And in ischemic stroke, that's when a thrombosis or an embolism restricts blood supply to the brain. It results in insufficient oxygen and glucose, which leads to a deficit in cellular homeostasis. You get energy failure, membrane depolarization, and excessive glutamate release. So uncontrolled glutamate release then leads to further membrane depolarization. It creates an excitotoxic positive feedback loop, which allows calcium to rush into cells across its concentration gradient. And that activates many calcium-dependent enzymes, such as phospholipases, proteases, as well as DNases, and they take cells apart from within. That causes necrosis in the infarct core. And then less severe energy failure leads to mitochondrial dysfunction, oxidative and nitrogen stress, DNA damage, and apoptosis in cells adjacent to the infarct core. And the net effect is this widening band of cell death that radiates out from the primary site of hyperperfusion. You get the release of damage-associated molecular patterns from dead and dying cells and acute inflammation. And together, those processes lead to the generation of a distinct infarct within hours of stroke onset. So I'm showing some images here of the pathology of what a stroke looks like in both the human brain as well as a rodent brain. So on the top, there's a stroke in the territory of the middle cerebral artery in a human brain. Underneath, there is a stroke in a rodent brain where we surgically occlude that same artery, the middle cerebral artery. So you can see that the pathology is in approximately the same place. And at the bottom, it's a human brain that's had a stroke in the territory of the middle cerebral artery, but it's a chronic stroke. So this is what the stroke looks like several weeks or months after the stroke. You can see that the stroke side of the brain is slightly smaller because there's been atrophy. And I'll talk more about the pathology of that stroke infarct later on. So in terms of prevalence, about 800,000 people suffer a stroke in the US every year. More than 15 million people suffer a stroke worldwide. And 1 in 4 people over the age of 25 will suffer a stroke in their lifetime globally. There's only 2 treatments at the moment. The first is treatment with tissue plasminogen activator that was approved in the 1990s. This is a thrombolytic. It's a drug that if you can get it to people within 4.5 hours, it can bust the clot that's causing the stroke. The other treatment, which was approved afterwards, is mechanical thrombectomy. So that has a treatment window of 24 hours. That's where you can use a corkscrew-like device surgically to remove the thrombus that's causing the stroke. But this is something that's only can be used in approximately 20% of patients. So these are the only two treatments available to help people suffering from stroke, but their time window is very short. Hypertension is the most prevalent risk factor for stroke, so it's very important that people know their blood pressure. And something that's going to have an impact, I think, in the next few years in terms of the prevalence of stroke is that there's been the development of watches that have blood pressure monitors in the wristband. So this means that it's going to be easier for people to know when they have hypertension so they can be identified and then it can be treated. So there was an article that came out a long time ago, back in 2006, and it was just drawing attention to the fact that even back then, there had been over 1,000 experimental treatments in acute stroke that had been tested in preclinical animal models. However, despite those many thousands of treatments, so it's probably closer to 10,000 today, how many preclinical studies there have been in rodent models of stroke. Despite that, there still aren't any treatments to help people recover from stroke. There's also been over 100 clinical trials for the treatment of stroke that have all failed. And these are clinical trials that have gone straight from the back of the preclinical studies that have targeted obvious things. They've targeted oxidative stress after stroke, they've targeted excitotoxicity after stroke, and they've targeted acute inflammation after stroke, but they've all failed. There's that one drug that's in use today, tissue plasminogen activator, to help bust the clot that's causing a stroke. But no other therapies have been developed since the introduction of TPA in the 1990s. But something that I think is really instructive is that TPA itself was only successful in 3 out of 11 clinical trials. So even delivering something as obvious as a thrombolytic to bust a clot that's causing ischemic stroke failed more often in clinical trials than it succeeded. So in order to help with the translation of preclinical stroke treatments, there's a group called the Stroke Therapy Academic Industry Roundtable, and they came up with a list of recommendations to maybe increase our odds of translating something successfully for stroke. And these are recommendations that would probably apply just as well to spinal cord injury and traumatic brain injury. And their recommendations are to make sure that your studies are always randomized and blinded, that before going to a clinical trial, everything is tested and verified in 2 or more labs. Things are replicated in a second species. A sex difference is considered. The route of administration is considered. And the time window and dose response is something that's thought about. The inclusion and exclusion criteria should also be predefined for a preclinical experiment, or at least we can do exploratory preclinical experiments, but before going to a clinical trial, make sure that there is another preclinical experiment that has been designed like a clinical trial. Also, conducting full power analyses. I know we all do this, but still, despite people doing their power analysis, things seem to be underpowered a lot of the time in preclinical experiments. And the inclusion of aged animals and animals with comorbid conditions such as hypertension, diabetes, and hypercholesterolemia. So there's no equivalent set of guidelines, however, to help clinicians overcome the obstacles that they face to get a stroke treatment through a clinical trial. So what I'm showing with the cartoon is that there's a valley of translational death in that in the preclinical world, we're oftentimes getting things that look like they're successful at improving recovery from stroke, but also obviously spinal cord injury and traumatic brain injury, but they're not passing the clinical trials that then they're moving forwards into. And obstacles that clinicians face in doing a clinical trial for stroke are that different patients are on different medications. So drug interactions through a trial need to be considered. Different patients have different comorbidities. There are many different types of stroke as well. For example, you can have embolic strokes in different territories, single strokes, multiple strokes, and non-embolic strokes. The state of the brain prior to the stroke is not the same in all patients. For example, the extent of white matter disease. The trajectory of recovery after stroke varies widely. And patient retention is also a major problem in any studies that are hoping to look at recovery after stroke, where you want to track people for 3 months or 6 months or a year. Another challenge is that there's no established biomarkers for stroke trials. That I think is going to change in the next few years, especially with neurofilament light becoming something that's more popular for tracking neurodegeneration in people after a neurodegenerative disease. And the other problem is that clinical trials are also probably nearly always underpowered because the primary endpoints lacks the sensitivity required to deal with all of the patient heterogeneity highlighted above. We have another problem with stroke research as well, and that is that there's this nihilism among the biggest US pharma companies for the development of treatments that help stroke recovery. So there are drugs being developed to help stroke patients, but most of the drugs in development are focused on secondary prevention. So for example, new and improved statins, or new and improved versions of tissue plasminogen activator. So new and improved thrombolytics. So when I was looking at all the pharmaceutical companies in the S&P 500, there was only one that I found, AbbVie, that has something that they're promoting in their pipeline that is addressing stroke recovery. The rest of the companies, there's nothing that goes beyond something that's just focused on secondary prevention or thrombolysis. And this is a really big shame simply because of the high prevalence of stroke. If 1 in 4 people worldwide over the age of 25 are going to have a stroke, the fact that very few companies are focusing on anything that can enhance the recovery means that there's a gap. Denialism is understandable, though. So billions of dollars have been lost in clinical trials that have failed. But hopefully, because there's extensive overlap in the inflammatory and recovery mechanisms in stroke, TBI, and spinal cord injury, then treatments that improve recovery in one of these diseases may have some efficacy in stroke as well. So I'm hopeful that as soon as something's developed for the treatment of TBI, spinal cord injury, or stroke, then it'll be something that can be applicable for all of these diseases. And I think as soon as one thing is shown to be successful, then I think pharma is going to suddenly turn its attention back to stroke and maybe invest more in TBI and spinal cord injury as well. I think that's going to happen in the next few years, but until then, there's definitely this nihilism in pharma for developing treatments that help with stroke recovery. Okay, so now I'm going to talk about the aftermath of stroke. So frequently after stroke, something that commonly is presented is that people have some paralysis of some sort. Maybe it's a facial droop, maybe it's paralysis in an arm or a leg. And while they have that paralysis, then something that happens is people are going to undergo some physical rehabilitation. But I've used a red box to highlight the chronic stroke at the bottom on the left-hand side there. Because while people are undergoing physical rehabilitation, they still have the problem of that they've got a chronic stroke infarct with an area of inflammation in their brain. Also, while patients are undergoing cognitive rehabilitation and physical therapy, about 1/3 of those patients will develop dementia within a year of their stroke. Now, in people, the cause of dementia after stroke is very difficult to determine, and that's due to the fact that most stroke patients are elderly. And so there's going to potentially be the coexistence of Alzheimer's disease, frontotemporal dementia, Lewy body dementia, non-large ischemic stroke vascular dementia, and dementia associated with Parkinson's. So my lab goal has always been focused on developing treatments that can improve recovery from stroke and reduce the risk of dementia after stroke, because our hypothesis is that chronic inflammation is a driver of dementia after stroke in some people. So we have this very, very simple overriding hypothesis for all the work we do in my lab, and that's simply that dementia in some people may be caused by smoldering inflammation in the areas damaged by the stroke. And that in all patients, even if you don't get dementia after stroke, that smoldering inflammation is a barrier to your recovery while you're undergoing physical rehabilitation. And so all we want to do is come up with treatments that can just put out the smoldering inflammation. And I'm using this word smoldering a lot because I don't want people to come away with the idea that you have a stroke and your brain is on fire with inflammation the weeks and months afterwards. I don't think that's the case. I think there is just this low grade, fairly mild, smoldering inflammation that persists. But the fact that it's persisting for weeks, if not months, is the problem. And also, I don't think there's anyone who would be happy, even knowing that there's smoldering inflammation that's persisting in their brain after stroke, if it's something that could be treated, but is not currently being treated. So here again is the pathology of a stroke infarct, chronic stroke infarct in the territory of the middle cerebral artery in a human brain. Here's another picture of a chronic stroke in the territory of the middle cerebral artery in the human brain. And it's unusual in terms of the consistency. It looks a lot like atheroma. And something that happens to the brain after a stroke is it undergoes this process of liquefactive necrosis. That's a very unusual process to happen in response to a sterile injury. If you look up liquefactive necrosis in Wikipedia, which I did several years ago, it was just kind of interesting that it said that the definition of liquefactive necrosis is it's a response to infection. It's something that happens in the lung, but it's also something that happens in the brain in response to stroke for unknown reasons. Now, eventually over time, the area of liquefaction is removed and you're left with a cystic space. But it's unknown in humans how long it takes for the process of liquefactive necrosis to run its course and for you to be left with a cystic space where previously you had brain. But what is evident is that around that cystic cavity, when you finally have it, you've had a lot of atrophy of the hemisphere or the brain regions around that area that was liquefactive necrosis. And also the title or the name liquefactive necrosis, and these are terms coined by pathologists. It's just kind of enlightening in that it indicates that maybe degradative enzymes are one of the problems of chronic stroke, in that for an extended period of time, maybe there's very high expression of degradative enzymes and they are beating up and causing further injury for weeks to the territory surrounding the infarct. I'm also now putting up an image, an MRI image of a stroke infarct. And the reason I'm doing this is just to highlight that, while this looks bad, the MRI image, that someone's clearly got a large stroke, something that I don't think is appreciated enough is that that light spot where you can see that stroke infarct is an area of liquefying brain tissue. And it looks almost too benign when you just see it in black and white like that. That if you could actually create a window and see what the brain looks like in an individual who's had a stroke, who's undergoing physical rehabilitation, and someone who has no drugs to support their recovery, and saw that they have this liquefying large amount of brain tissue adjacent to all the brain regions that weren't hit by the stroke, there'd be this increased sense of urgency to test and develop treatments that target chronic inflammation after stroke. And in all the research that I've done, I haven't come across any trials, in fact, where someone has received a drug, an anti-inflammatory drug, for 3 months or 6 months after they've had their stroke. There have been clinical trials for stroke that I'm aware of where people have received an anti-inflammatory medication for a few days, but nothing that I found where people are receiving an anti-inflammatory medication for weeks and months. So this is something that's happening in the brains of people who suffer a stroke, and right now it's not treated. So the initial research questions I had for my lab when I started it back in 2013 were, again, very simple questions. The first was, how long does the inflammatory response to stroke last? How long does it take for that dead tissue, that liquefying tissue, to be removed? How well is it segregated from the rest of the brain? Because scars form to segregate dead tissue from the rest of the brain, to segregate the inflammation. But how well does that scar segregate the damaged area from the rest of the brain? And also, why does the brain undergo liquefactive necrosis after stroke, whereas the heart doesn't undergo liquefactive necrosis following a myocardial infarction? What's different about the brain? The stroke model I employ is a model of distal middle cerebral artery occlusion plus hypoxia. And I have the image of a mountain because we placed mice in a hypoxia chamber containing 8% oxygen, which is equivalent to putting a mouse at the peak of K2, which is the second highest mountain in the world. And to induce the stroke, we drill a hole in the side of a mouse's skull, and then we cauterize the distal region of the middle cerebral artery. Now, over the years, a challenge to getting funding and publications, rightly, is that reviewers will say that giving a mouse a stroke and then putting it at the top of a mountain is not physiologically relevant, except very few people who have strokes at the top of mountains. So it's not a very useful model for stroke because you're doing that strange thing of reducing the amount of oxygen. The reason we're doing it is to make the stroke damage much larger, and that makes some other experiments we do much easier. But I'm going to talk now a little bit more about how we came to develop this model of stroke. And to do that, I'm going to begin by introducing someone called Thomas Willis, who was a 17th century anatomist and neurologist. And in 1664, he published a book called On the Anatomy of the Brain. And it's one of the best examples of 17th century anatomical achievement. And in this book, he has images of the base of the brain. And at the base of the brain, there's a structure that was named after him. and it's the circle of Willis. And the circle of Willis is a circle of arteries on the base of the brain, of which a particular feature has been an obstacle, I think, to the development of stroke treatments for decades. So earlier I put up all the different STAIR criteria that have been set to try and help with the translation of stroke treatments. But something that isn't included in their criteria and isn't talked about too much is a problem with the circle of Willis that's hindered research into the chronic consequences of stroke. And the problem is that although the circle of Willis connects the carotid and vertebral arteries, in some mice, it doesn't. So on the left-hand side, you can see an anatomical diagram of the circle of Willis. It's connecting the carotid circulation with the vertebral circulation, because going up to your brain, you've got the two carotid arteries and you've got two vertebral arteries. And they're connected by two vessels called the posterior communicating arteries. But in black 6 mice, C57 black 6 mice, which is the strain that I've used predominantly for my career, and a lot of other people use them too, because you can get transgenic mice on this background, and a lot of research is already established in the mice. But in this mouse strain, one or both of these posterior communicating arteries is often missing. This is what the circle of Willis and the posterior communicating arteries look like in a C57 black 6 mouse. Now, when we took a look to see how many black 6 mice actually have an intact circle of Willis, what we found is that only about just over 1/3 of them had an intact circle of Willis. Most black 6 mice either have one posterior communicating artery absent, or they have both posterior communicating arteries absent. And this is something that Barry McColl at the University of Edinburgh has also looked at, and he's found approximately the same variability in circle of Willis anatomy. Now, mice can survive normally with an incomplete circle of Willis, and this happens in people too. So there's a lot of people out there who don't have a complete circle of Willis, that you're missing a posterior communicating artery. But a mouse can survive quite normally when it doesn't have a complete circle of Willis. And when you have a complete circle of Willis, what happens is the territory fed by the middle cerebral artery receive blood that's coming up from the internal carotid artery. And when you have that complete circle of Willis, the territory fed by the posterior cerebral artery receives blood coming up from the vertebral arteries, as you can see here. Now, when you're missing a complete circle of Willis, what happens is blood can perfuse the territory fed by the middle cerebral artery and the posterior cerebral artery simply from the internal carotid artery, as you can see here. So it's not a problem if you don't have a stroke, but it matters for stroke research. And the reason for that is that for the most common model of stroke, what happens is a surgeon will take a suture, do an incision in the neck region of the mouse, and thread a suture up the carotid to the bifurcation point of the middle cerebral artery and the anterior cerebral artery. And you keep advancing that suture till it reaches that bifurcation point. You won't be able to advance it anymore, and it prevents blood from perfusing the territory fed by the middle cerebral artery. Now, with an intact circle of Willis, this results in a stroke infarct in the territory supplied by the middle cerebral artery, this territory. But when you have a missing posterior communicating artery on the ipsilateral side, then it results in an infarct in the territory supplied by both the middle cerebral artery and the posterior cerebral artery. And this is a problem for stroke research, because these mice will often die because their stroke is so severe within a few days of doing the stroke procedure. Or another problem is it just causes a lot of variability in the stroke model. So here's an example of a mouse, a black 6 mouse that undergoes that stroke procedure that had an incomplete circle of Willis. This is a very, very large infarct. You'd be lucky to have this mouse survive past 7 days, so you can't really do a investigation of chronic inflammation after stroke. This is now a black 6 mouse that's had exactly the same surgical procedure. So it's a genetically identical mouse, the same procedure happened, but you can see that the damage caused by the stroke is very, very different. So this mouse probably had a complete circle of Willis. So this, I think, has been a very big problem for studying the chronic consequences of stroke and completing long-term behavioral studies. And I think it's also a reason why a lot of stroke studies including my own, are just chronically underpowered. So in order to study the chronic consequences of stroke, during my postdoc, we spent some time designing a new stroke model. So that's where we knew that if you do the distal middle cerebral artery occlusion model of stroke, then you can bypass the problem of whether or not there's the presence or absence of a posterior communicating artery. So basically, whether you have a complete or an incomplete circle of Willis, you will always maintain circulation to the territory fed by the posterior cerebral artery when you occlude the distal medial artery from the distal region by doing craniectomy. And I'm just illustrating that here. So in an intact circle of Willis, you can get blood going to the territory fed by the posterior cerebral artery. And then on the other side, you can see where there's an incomplete circle of Willis, we're missing that communicating artery. You can still get blood perfusing the area fed by the posterior cerebral artery. It's just coming from the carotid circulation now instead of the vertebral circulation. However, if you do a distal cauterization of the middle cerebral artery, what we find is that the damage caused by that stroke is very, very small. You've got a small infarct centered on the barrel cortex, which is the bit that receives input from the whiskers. So mice have numbness to the side of their face, but they don't have any other behavioral deficits. So it's very difficult to look at recovery if you don't really have any discernible behavioral deficits. And also the amount of tissue infarcted means you don't have a lot of tissue to work with, which is problematic for modeling the chronic consequences of stroke. So in order to make the stroke damage more severe, we tried, in addition to cauterizing the distal region of the middle cerebral artery, we targeted additional blood vessels by tying sutures around one or both carotid arteries. And then we also tried placing mice in a hypoxia chamber and lowering the oxygen concentration to 8%, where it's normally about 21%. And what we found was that when we tried doing additional surgeries and tying a suture around one or both carotid arteries, we didn't get around the problem of variability in circle of Willis anatomy, and there was still a lot of variability with those surgeries. It took an hour and a half to do each individual surgery, so you can do 4 or 6 animals in a day. But when we used the hypoxia chamber, we found that we no longer had to deal with the problem of circle of Willis anatomy. We could do 30 to 40 surgeries in a single day, and it tripled the size of the infarct. So it had a lot of advantages. Importantly, because we made the infarct bigger, mice have an immediate motor deficit when we have them traverse a ladder. So they have a fault on the ladder test, and they develop a delayed deficit in cognition. So this is data from the Y maze test, where at baseline mice can navigate a Y maze alternating through the different arms at a level approaching 60% before they've had a stroke, which is what you would expect a normal mouse to be able to do. It means it can remember where it's just been. A week after the stroke, it can still do that test, but 7 weeks after stroke, they developed a problem with their memory. So behavior is something that everyone who's done it in mice knows it struggle with sensitivity. It's not necessary when you get a good result, anything that you think you can expect to find again when you repeat the experiment. So whenever you see behavior data, the other thing that you're going to want to see is the pathology to back it up. Evidence that there has been a problem developed in the brain that would corroborate the behavior that you see. So we also find that with this stroke model. So what we find is that a week following stroke, you can see that there is an infarct in the territory supplied by the middle cerebral artery, but otherwise both hemispheres of the brain look very symmetrical. When you advance to 7 weeks after stroke, now, however, there's a lot of asymmetry in the brain. There's been evidence of cortical atrophy, swelling in the hippocampus on the stroke side of the brain looks a lot bigger, and there's ventricle enlargement, probably due to atrophy of the stroked hemisphere. We also find when we look at neurofilament light that there is consistently an elevation in neurofilaments in the blood of these mice in the weeks after stroke. Now, acutely, 24 hours after stroke, there's a massive release of neurofilament light into the plasma, which you would expect. It goes down fairly rapidly, but even as late as 7 weeks and 6 months after stroke, it's still mildly elevated compared to naive mice. So we think there is ongoing neurodegeneration for several months after stroke. Just as importantly, variability and mortality are low with the stroke model that we're using. And here's a comparison of infarct size and variability in each of the stroke models that are being used today, with the exception of a photothrombotic model of stroke, which we haven't included here. But I'm just using an arrow to highlight the stroke model we're using gives you the best bang for your buck in terms of the size of damage you're causing and the smallest amount of variability. And it also has the advantage that you can do a lot of mice at once. So I know this was a lengthy digression, but I just wanted to draw attention to this model of stroke we're using, because as far as I know, there aren't any other labs that are using this model of stroke. It's given us tremendous advantages. And so I'm trying to generate, number one, provide an explanation for why when I visit other places, everyone always says they've had this massive amount of variability in their stroke model. So I wanted to draw attention to the circle of Willis and inconsistency in anatomy as a potential explanation for that, but also show that we have an alternative model that I think is quite valuable. Okay, so now going back to the actual science. So how long does the inflammatory response to stroke last? After developing that model, we did a very basic experiment. We just took mice, we gave them the stroke model, and we started harvesting tissue at these different time points to try and identify how long inflammation lasts after stroke. And I'm going to show you a summary of that data now. So in the first week after stroke, the first thing that you see is that in the first few days, monocytes from the periphery infiltrate the infarcted tissue, they mature into macrophages, they join with surviving microglia in the phagocytosis of dead tissue. And you see this microglia and monocyte response, not just in the infarct, but also in the area of axonal degeneration projecting from the infarct. Now, the immune cells that are in the infarct, as well as the area of axonal degeneration, are producing cytokines. They're producing acutely high levels of IL-6. They're also producing TNF-alpha, IL-1 beta, and interferon gamma. An important experiment we did early on was we sought tissue, human tissue, from patients that had an acute stroke. And we just compared the cytokine response in black 6 mice and humans to make sure that there is somewhat of an equivalent cytokine response in both mice and humans. We also did this in another strain of mouse, BALB/c mice. And what we found is that there is pretty good overlap in terms of acute inflammation in both mice and humans. There's other aspects of inflammation which are quite different, but at least in terms of the cytokine response, it seems to be similar. Now, in the first few days after stroke, there are some, but actually very, very few T lymphocytes and B lymphocytes in the infarcted tissue. So here, this is representative images of a B cell stain, a pan B cell stain using BT20, and a T cell stain using CD3. And what's notable is the absence of any staining. In the infarct, you don't really see any T lymphocytes or B lymphocytes at 1 week post-stroke. What you do notice, though, a week following stroke is that new blood vessels begin to form in the infarct. So this is shown with an H&E stain. So looking just at the area that we infarcted, you can see that there's this network of new blood vessels that start forming in the infarct. And at the later time points, you can kind of see what they're doing. They're delivering the large numbers of immune cells necessary to help clear and eat up all the tissue that was destroyed by the stroke. Now, we've quantified the formation of those blood vessels using CD31, which is an endothelial marker. And you can see that there's a very rapid formation in the first week. And then as the infarct is being eaten away and it's turning slowly into a cavity, those blood vessels get resorbed. So they're only there for as long as they're necessary to deliver the immune cells to eat the dead part of the brain. However, for as long as they're there, they appear to be leaky. We know that by injecting Evans blue into the circulation of mice. With an intact blood-brain barrier, the brain should not turn blue. 24 hours following stroke, the infarct turns very blue. There's a lot of restoration of the blood-brain barrier in the first 7 days, but even as late as 7 weeks, the area where we see those new blood vessels still turns blue. So those blood vessels are chronically leaky. And when you look at them down the microscope with an H&E stain or a CD31 stain, they just don't look very good. They look a lot like blood vessels that form in a tumor. They look like it's just been it's a tortuous, ugly emergency response of neovasculogenesis, probably to just deliver immune cells to eat away the dead material, and then they'll get resorbed over time. Now, another reason why they're leaky is that the new blood vessels are not forming anything like a new intact neurovascular unit. If you do a GFAP stain for astrocytes, what's notable is that the infarct doesn't have any astrocytes. The astrocytes that were there have been killed by the stroke. a border forms, but within the infarct itself, where you have those new blood vessels, there's no astrocytes to put end feet on the blood vessels and form anything like a neurovascular unit. Using an NG2 stain as well, we don't see any parasites on those new blood vessels that rapidly form within the territory of the infarct. And when we look for degradative enzymes, matrix metalloproteinases, we find very high levels of MMP3, 2, and 9 within the infarct. And these are the degradative enzymes that are probably mediating the liquefaction of the dead part of the brain. But matrix metalloproteinases also degrade tight junctions. So even if tight junctions were trying to form on those blood vessels, these degradative enzymes would maybe prevent them from being established. And finally, when we did a Claudin-5 stain, we found that in the peri-infarct location, we do see that there's tight junctions, as evidenced by that Claudin-5 staining in the peri-infarct location, but on the blood vessels in the infarct, we don't really see any evidence of tight junctions. Okay, so in the first week or 2 weeks following stroke, the other thing that happens is the infarct becomes to be compartmentalized by an astrocytic glial scar. So here's some imaging of a GFAP stain, just to show you what the glial scar looks like. Astrocytes bundle together, they become hypertrophied, and they orient parallel to the lesion. We did some scanning electron microscopy to look at the ultrastructure of the infarct and the glial scar. And we stained the glial scar in this magenta color. And we have the infarct shown in yellow, and then the peri-infarct white matter is shown in the blue color. So you can see there's just this interwoven hypertrophied bundle of astrocytes that form the glial scar. Now let's advance to week 7. So as I've shown before, there's this further neurodegeneration that happens in the weeks after stroke. In concordance with that, as the hippocampus starts swelling, we find that mice develop a deficit in long-term potentiation. So at week 7, you can see now that the stroke mice are diverging from the control mice in their ability of the hippocampus to undergo long-term potentiation, and it looks even worse at week 12 than it did at week 7. And when the degeneration is severe enough, mice develop a delayed memory deficit. So here I'm showing again the Y-maze data showing that mice develop the appearance of a delayed memory deficit on the Y-maze. Now that progressive neurodegeneration the weeks after stroke correlates with the appearance of T and B lymphocytes in the infarct. So I showed earlier that you don't really see much in the way of T and B cells in the brain a week after stroke. Now at the top, you can see that at 7 weeks, however, there's thousands of B lymphocytes that are now in the infarct. Underneath that image, there's a time course of T cell infiltration. And what's really interesting about this is that there are even more T cells at 7 weeks than there were at 4 weeks, that it's just progressively getting worse, the T cell infiltration into the brain. This is the time course of the B cell infiltration. It's very similar to the time course of the T cell infiltration. It's just getting progressively worse over time. Now, glial scars, they form a great barrier that's trying, or one of its functions is to segregate the area of chronic inflammation from the rest of the brain. However, they're permeable barriers. We know that because we injected a fluorescent dextran into the infarct, and then it sacrificed mice a few hours later, and we injected this dextran into the infarct 7 weeks after stroke, and found that the dextran can cross the glial scar and get to the other side of the brain. This is important because it means that antibodies that are being produced by the B cells, and there's hundreds of plasma cells in the infarct late after stroke, have an access point to get into the surrounding brain tissue that's atrophying. They can just cross the glial scar. Not only that, but all the molecules, plasma proteins leaking out of those leaky blood vessels can cross the glial scar and get into the surrounding tissue. And all the cytokines that are chronically present and degradative enzymes chronically present in the infarct after stroke, while being restricted by the glial scar, are not impermeably restricted by it. They can saturate, permeate slowly, insidiously for many weeks and months into the surrounding tissue. And we think this is a cause of the atrophy in the brain. This is what the brains look like by MRI 7 weeks after stroke. Again, you can see that there's an increase in the amount of T2 signal adjacent to the infarct. This correlates with the enlargement of the hippocampus, which is why I think it's swelling of the hippocampus. The inflammation is just causing the hippocampus to swell. So back in probably around 2013, 2014, we turned our attention to B cells first, thinking that because there's this flood of antibody and thousands of B cells accumulating in the infarct after stroke, and we wanted to see whether or not if we get rid of the B cells, maybe we can improve recovery. And there's somebody called Philip Popovich who does a lot of spinal cord research who had already shown that B cells impair recovery after spinal cord injury. And so we wanted to see if we could take a similar approach to him to improve recovery after stroke. To test that hypothesis, we just ordered B cell deficient mice. They're called mu-MT mice. They don't have any more mature B lymphocytes. The first thing we did is make sure that they have a similar size stroke to a wild type mouse in our stroke model, which they do. Then we just validated that they don't have any mature B cells, which they don't. There's no B cells that turn up in their infarct after stroke, and they can't make antibody. So there's no antibody that permeates the infarct after stroke. And what we found in these mice is that they didn't develop the delayed deficit in long-term potentiation, and they didn't develop a delayed deficit in the novel object recognition test or the Y-maze test. So this was great. Back in 2015, we published a paper where we thought we'd done this wonderful thing of showing that if you can target the B cell response to stroke, you may be able to improve stroke recovery. However, since realized that the B cell response to stroke is not an optimal therapeutic target for a variety of reasons. One is that it's strain specific. So when we looked at another strain of mouse, BALB/c mice, we didn't see that the same thing happens in those mice. They have much less severe B cell infiltration. And we looked at human postmortem tissue. They don't have a similar B cell infiltration. Humans don't have a similar infiltration after stroke either. I think that it's likely that in some people, there's a B cell response elicited by stroke, but I don't think it happens in all people, which would make a clinical trial very difficult because you'd have to identify those patients first. Also, ablating B cells could potentially be immunosuppressive in a population already at risk of infection after stroke. And finally, we've since found that the B cell response to stroke is very heterogeneous. It consists of different types of B cells, some of which may be assisting with recovery. For example, something that was very strange is that when we took a T cell knockout mouse, a CD4 knockout mouse, and when we took an MHC2 knockout mouse, so they can't present antigen to CD4 T cells, what we found with the B cell stain is that there's still thousands of B cells that migrate to the stroke infarct at 7 weeks after stroke. So what this means is that a large part of the B cell response to stroke is happening by a population of B cells that aren't requiring helper T cells for their activation. So if anyone's had an immunology course, one of the things that you learn about B cells is that they require help from helper T cells to be activated. Another much smaller part of the curriculum for immunology classes is that there's actually a population of B cells that can be activated independently of any T cell help whatsoever. And they'll either be activated because their toll-like receptors are receiving so much stimulation, you don't need the BCR to receive an antigen, or they'll be activated because they're seeing so much antigen that it cross-links the T cell receptors together, and then it bypasses the need for T cells or toll-like receptor ligation. And what we found by looking at B cell responses in the absence of helper T cells and in the absence of toll-like receptor help as well, is that a large part of the B cell response to stroke looks like it's happening, as can be shown in the bottom diagram. It's happening in response to what's known as a TI2 antigen, or rather it's happening in response to antigens that are cross-linking B cell receptors together, and it's happening to B cells that produce what's known as natural antibodies. And these are B cells are not part of adaptive immunity. They're part of innate immunity. They come from the fetal liver, produces them. They're associated with mucosal immunity in the gut. And one of the things that, or one of the antigens that they bind to is lipids. So we're not sure what the antigen is yet, but our hypothesis is that the B cell response to stroke, a lot of it could be in response to lipid antigens. Okay, so the chronology of inflammation I've shown so far is that you have this tortuous network of new blood vessels forming in the infarct. It gets segregated from the rest of the brain by a glial scar, but not perfectly. You have a lot of macrophages derived from monocytes turning up into the infarct. In the days after stroke, they're drawing in T cells and B cells. The T cells and B cells are then producing antibodies and cytokines that are leaking into the adjacent tissue, which we think is mediating atrophy and chronic neurodegeneration. Okay, so now turning to foam cells. So this image is something that immediately made us think that infarcts basically are just a sea of foam cells. That's what it looks like. It looks like a bubbling mass of foam cells. We did some transmission electron microscopy and saw the same, and it gave us that same sort of like impression that foam cells are a better target than going after adaptive immunity. When we've looked at foam cells, we found that they're consistent in every strain of mouse we've looked at, and they're also a consistent finding in all the human postmortem tissue we've looked at. Foam cells are cells that also accumulate cholesterol crystals within them, and cholesterol crystals are bad because they can activate something called the NLRP3 inflammasome and drive the production of cytokines that can then draw in other immune cells as well as themselves be neurotoxic. Now, we wanted to understand whether or not the accumulation of foam cells was typical for an inflammatory response to a sterile injury. To do that, an experiment we did was we gave some mice myocardial infarctions and some mice our model of stroke, just to see whether or not this is something that happens to any tissue following an injury. So this was in collaboration with someone called John Canhelis at the University of Arizona. His lab induced myocardial infarctions for us, and then we killed mice at different time points. And the histology here is to show you that the size or the extent of damage is fairly equivalent with our brains and our hearts. I mean, these are large myocardial infarctions that we're causing, and we're causing strokes that are destroying about 1/4 of one hemisphere. What we found was that in the heart, we found that there is a collagen scar that forms within about 4 weeks of the injury. Whereas in the brain, you can see some collagen being laid down at 4 weeks following the injury, but you don't see a dense collagen scar. Also, at 4 weeks, you see infiltration of lots of T cells and B cells in the brain that doesn't happen in the scar. And there's a good amount of CD68 staining in the brain that doesn't happen in the heart. And CD68 is a scavenger receptor and one of the markers for foam cells. Now, instead in the brain, while it took 4 weeks for the heart to basically get to an end stage or a resolved stage of inflammation, in the brain, it took 24 weeks or 6 months. So at 24 weeks, we found that the T and B cell infiltration was largely vascular by that stage. It looks like the tissue itself no longer had an adaptive immune response. There wasn't as much CD68 staining anymore, but importantly, the collagen. So we had a collagen scar in the brain that looked like the one that turns up in the heart in 4 weeks, finally there by 24 weeks. So the inflammatory response is slower to resolve in the brain than it is in the heart. And one of the reasons we set up the experiment this way is that we knew obviously that the brain has a lot of myelin, the heart is not. The brain is lipid rich, the heart is protein rich. So it's just to show that when you take these two different organs with the different consistencies, that to lend credence to the idea that your immune system struggles more to process lipid-rich debris than it does protein-rich debris. Also in the brain, we found that there was a second wave of inflammation that started increasing between week 4 and 8 after stroke. That second wave of inflammation doesn't occur in the heart. So in the heart, there is an ischemic insult, acute inflammation, and then just resolution until you have a collagen scar. In the brain, there's acute inflammation, there's resolution, but then inflammation goes back up again. And this is protein data done by multiplex immunoassay on the infarcts from the brain and the heart. Now, that second wave of inflammation correlates with the formation of cholesterol crystals within the brain as well in the infarct. So we quantify cholesterol crystals by using a polarized light microscope because they refract light. And we find that there are more cholesterol crystals at 8 weeks than there were at 4 weeks. And their appearance correlates with that second wave. The development of cholesterol crystals also sort of like coincides with the accumulation of the T cells and the B cells and with the accumulation of the foam cells. In terms of where the cholesterol is coming from, we think it's coming from myelin. Here's some evidence that it may be coming from myelin as well. So when we look at the infarct at 7 weeks after stroke, it stains intensely with oil red O. It also stains heavily for sulfatide. And we see an abundance of sphingomyelins in the infarct at 7 weeks following stroke, or a concentration of them compared to the equivalent area of the brain on the contralateral side. Jay Lee has also done a lot of this work sort of ahead of us in spinal cord injury. So his papers have definitely guided us in terms of what we're looking at. So there's the paper as well that supports that myelin catabolism may be one of the drivers of foam cell formation that comes from the work that he's been doing. And so our overarching hypothesis at the moment is that cholesterol processing is driving smoldering inflammation after stroke. To test that hypothesis, that's where we've been testing an agent called 2-hydroxypropyl beta-cyclodextrin that has a high affinity for cholesterol to see if it could chelate excess cholesterol and other lipids that are generated by myelin breakdown and then do the job of macrophages for them. Now, this is something that's present in many pharmaceutical products, or cyclodextrins are. It's many different types of cyclodextrins are FDA approved as excipients or vehicles for other drugs. This is what the molecule looks like. It's a sugar. So it is a ring made of 7 glucose rings that come together to form a barrel-like structure. And the simple idea of all we're trying to do is use it as something that can just maybe encapsulate lipids derived from the breakdown of myelin, maybe cholesterol, maybe oxidized forms of cholesterol, so that it does the job of macrophages for them. And that's just illustrated here as well. We're delivering this molecule, this barrel-shaped molecule, so it can do the job of a macrophage, so a macrophage has the opportunity to not become overwhelmed and not turn into a foam cell. But cyclodextrin has also been reported in a paper that came out in 2016 by Zimmer et al. That it can reprogram macrophages. So this paper that came out in Science Translational Medicine showed that when you deliver cyclodextrin in a model of atherosclerosis, the macrophages start making more ABCA1 and ABCG1, which are cholesterol exporters, and they do that through an upregulation of the liver X receptors. So potentially cyclodextrin could also not just be a dumb molecule that entraps cholesterol, but maybe it could be doing some sort of transcriptional macrophage reprogramming. This again is just an outline of what we're trying to do. We're trying to prevent myelin debris for overwhelming macrophages by delivering cyclodextrin in the hope that it can chelate excess lipid, as well as maybe do something directly to macrophages as well to upregulate the LXR. In order to test this drug, we only began treating mice a week after stroke. We wanted part of the brain to be completely destroyed by a stroke. Then we waited a week so we could help with the cleanup by delivering large quantities of cyclodextrin 3 times a week for 6 weeks. And this is what we found. It works profoundly well at dampening the inflammatory response to stroke. So Danielle Bechtel in my lab is a graduate student who's been spearheading this project. And it's very difficult to be blind in these studies. As soon as we look down the microscope with different immunostains for immune cells, it's fairly obvious that there's something different about some of the mice. They then turn out to be the mice treated with cyclodextrin, but it appears to prevent the infiltration of B cells and T cells, and it prevents plasma cell formation in the infarct as well. It also, at 7 weeks following stroke, strongly reduces the production of degradative enzymes. These are the enzymes that are probably mediating some blood-brain barrier permeability, as well as the enzymes that are breaking down the dead part of the brain in that process of liquefaction necrosis. We also find by looking at the RNA in the infarct that there is an upregulation of ABCA1, which is downstream of the LXR, in the mice that were treated with cyclodextrin compared to with the vehicle-treated mice. We also see a decrease in transcripts associated with adaptive immunity and an increase in transcripts associated with innate immunity. So to summarize the pathway analysis of what we find in the cyclodextrin-treated mice, we find that the drug is reducing inflammation, but it is improving lipid metabolism. We also find that CFOS, a transcription factor, synapse-associated transcription factor, we find that CFOS staining is increased or preserved in the cyclodextrin-treated mice compared to the vehicle-treated mice. And we find that there is a preservation of neurons in the striatum and thalamus. There's a reduction in hippocampal edema. There is an improvement in the Y maze at 7 weeks after stroke compared to vehicle-treated mice. And there is a difference in how mice are responding in the light-dark transition test after stroke as well in the cyclodextrin-treated mice. So we think it's something that's not only dampening inflammation, but is also improving recovery, and it's doing that by preserving neurons and neuronal function. Gene set enrichment analysis also showed that there was the preservation of pathways or gene sets associated with neuronal function activity in the peri-infarct location after stroke. And so for this project, our next steps are, we want to see if there's more neurodegeneration in comorbid models of stroke, so looking at models of hypertension and obesity, as well as ApoE4 mice. This is going to make our model more translatable. So essentially, we need to follow the STAIR criteria before we can advance it to a clinical trial. We also need to find the lowest effective dose of cyclodextrins and determine the optimal frequency of administration, because right now we're probably doing some deleterious things to the brain with the massive amounts of cyclodextrin we're doing. But right now, we're just focused on looking at the inflammation and the health of the neurons immediately adjacent to the infarct. And we're going to have to look at other brain regions and the impact on cholesterol homeostasis later on. We are currently testing it in females, as well as a second stroke model and a model of spinal cord injury. So we're paying particular attention to the STAIR criteria. And we're trying to develop a panel of blood biomarkers that we can use to monitor treatment efficacy, because that's what we'll want to use in the patient population, as well as come up with a new neuroimaging technique that we can use in a patient population. So I'm going to spend a little bit of time talking about this new imaging modality that we're developing. So we've got a little bit of data that we got fairly recently that suggests that something that we may be able to image is lipid metabolism in foam cells after stroke. So foam cells clearly have taken in a lot of lipid, and maybe on their path to becoming foam cells, and maybe while they are foam cells, it's likely that they are trying to metabolize some of that lipid that's overwhelming them. And so we got the idea of wondering whether or not, if that's the case, well, maybe that's something that could be imaged using PET imaging. So everyone here has probably heard of glucose imaging of the brain, where you can look at glucose imaging to look at brain function. Well, you could also potentially maybe do fatty acid imaging of the brain. And if successful, maybe that would be a way of imaging foam cells that infiltrate the brain after stroke, and then we could look at cyclodextrin efficacy. So to answer that question, here's just an overview of beta oxidation of fatty acids. The only thing I want you to look at is the molecule I've highlighted, or acyl carnitine. So acyl carnitine is something that needs to be added to a fatty acid in order for it to enter the mitochondria so that it can enter the TCA cycle. So the beta oxidation of fatty acids requires acyl carnitine to be added to a fatty acid. So we did an experiment where we did a time course of metabolomics, just looking at metabolism in the brain at these different time weeks after stroke to see if we could see evidence that fatty acid oxidation goes up in the brain after stroke. And then at one time point, 4 weeks, we did spatial transcriptomics to identify where an increase in beta oxidation was happening after stroke and in which cells. What my postdoc found is that in this heat map, what you can see is all of these acyl carnitines are elevated in the stroke brain compared to a naive brain going out to 4 weeks after stroke. So in accordance with the development of foam cells in the brain after stroke, we are seeing an increase in acyl carnitines in the brain that are conjugated to all these different fatty acids. So we think this is a signature that the beta oxidation of fatty acids goes up in the brain after stroke because immune cells are trying to incinerate the garbage myelin debris that they've taken up. So this was global metabolomics of the ipsilateral side of the brain. So we don't know what cells are doing this or where it's happening. So in order to pinpoint whether or not this is indeed happening in the myeloid cells, the microglia and the macrophages, and in the infarct and the area of axonal degeneration, that's where we turn to spatial transcriptomics. And we did find that in the heat map that says neurons, what you can see there, you can see that the blue, the darker blue, is in the transcripts associated with glycolysis. So this is just showing that, as everyone probably knows, neurons metabolize glucose for energy. Astrocytes, on the other hand, do actually metabolize fatty acids for energy. So 20% of your brain's metabolism is through astrocytes metabolizing fatty acids. When it comes to the microglia, they also use glucose for energy. But in the area of axonal degeneration, the TH, the thalamus, on the ipsilateral side of the brain, we found that there was a change. There was an increase in fatty acid oxidation and a decrease in glycolysis. So this is now enabling us to sort of pinpoint that the increase in fatty acid oxidation that happens after stroke compared to a naive brain looks like it's being driven by microglia in the area of axonal degeneration. And then when we looked in the infarct, we also saw that the TMEM119 positive cells are also increasing fatty acid oxidation in the infarct after stroke. So it's at an early stage, but we are hopeful, so we're gearing up to try this, that a radio-labeled with fluorine-18 fatty acid, palmitic acid, when we inject it into a mouse, we're hopeful that it's going to reveal the area of the infarct. And then we're going to give cyclodextrin and see if that can reduce it. And in this way, we're hoping in parallel to develop an imaging modality to be able to see foam cells that would be useful for a clinical trial, because otherwise we have a good target, but unless we have clinical endpoints that are going to be practical in the real world, with just like TPA going to fail at the clinical trial that we embark upon. So here's just an example of where PET MRI with a radio-labeled fatty acid has been used to image the heart before, but it's never been used in the brain. So we'll use the heart as a positive control. So when we deliver our palmitic acid that's been radio-labeled, we'll know if it works. If the heart turns up, we get a signal. And then there may be a signal in the brain because there's 20% of the energy comes from astrocytes burning fatty acids, but we're hoping that the infarct is going to turn darker. But we're not quite there yet, but we hope to get there in the next few months. So to summarize, there is a chronic inflammatory response to stroke. We think it's due to overwhelmed lipid processing, but in the stroke world, we're behind the spinal cord injury world in appreciating that this is something that's a driver of chronic inflammation. Foam cells are maladaptive and dysfunctional. They're present in the brain for months after stroke. And they're present in all humans and all the mouse strains that I've looked at. So they seem to be a common target. Stroke also causes a tortuous network of blood vessels to form in the infarct in the days after stroke. They persist for months. They're an area of chronic blood-brain barrier compromise. I think they should be a target in addition to foam cells. So in a combinatorial therapy, but they also mean that cyclodextrin should be able to get to our infarct because the blood-brain barrier is compromised in the region we want to deliver cyclodextrin to. And the current therapeutic approach we're investigating is the administration of cyclodextrin to treat foam cells, as well as concurrently with doing neuroimaging with radio-labeled fatty acids to see if we can image their formation and image the efficacy of cyclodextrin so we have something that would be useful for a clinical trial. And I know I went over, so I sped up a little bit there, but that's everything. And I just want to acknowledge, obviously, everyone in my lab who's been working this project. Danielle Bechtel's been generating most of the data I've shown today. And Sana Lopi, my postdoc, is gearing up to work on the PET imaging study. And with that, I'll take any questions.
Thank you. That was a really nice talk. I know you mentioned that you looked at the heart in this model. We've done some work on brain injury and lung disease and TBI. And I know with the model using hypoxia, which is a big cause of lung injury, have you looked at the lungs in these mice? And if so, how this, you think contributes to with the, a brain lung axis that's being researched more these days, if you think this contributes maybe to any of the deficits you're seeing in your mice?
It's a great question, and we haven't looked at the lungs yet. No. The lungs are so important for stroke research as well. So post-stroke pneumonia is one of the leading causes of death after stroke. Stroke causes animals and people to be immunocompromised. And so we're starting to look at the lungs with some collaborators, but we haven't looked at the lung yet. But there is something else interesting about the lung that I've not looked at. And that is that, so the lung undergoes liquefaction necrosis as well after injuries. And there's, in black 6 mice, a big B cell response to injury in the lung that doesn't happen in the heart following a myocardial infarction. So there's something different or similar about inflammation in the lung and brain that's different about inflammation probably in the heart and kidney. And it makes me wonder if lung surfactant, which is a high lipid content, may also be one of the reasons why you have maybe overwhelmed lipid processing following a lung injury. But I've not really done much work in the lungs yet.